Hello, our names are Abby, Ericka, and Simran, and we are researchers as part of the Big Data and Biology Lab. Disorders are conditions that alter one's emotional state and interfere with daily life. The two most common mood disorders are major depressive disorder, characterized by prolonged sadness, and bipolar disorder, characterized by periods of depression and mania. The National Institute of Mental Health has estimated that approximately 1 out of every 5 adults in the U.S. experience any mood disorder at some time in their lives. Around 1 million people commit suicide. 1/2 to 2/3 of these suicides are completed by those with mood disorder. Based on the observed connection between suicide completion and mood disorders, the goal of our research is to study suicide and mood disorders at a genetic level to better understand this relationship and the mechanisms involved. We analyzed RNA sequencing data from the brains of 73 postmortem donors. RNA was extracted from specific regions of the brain involved in regulating emotions and reduced in volume in people with mood disorders. Each sample had information regarding diagnosis of mood disorders, suicide completion status, and other important characteristics.
Our analysis consisted of finding differentially expressed genes and gene networks in samples with and without mood disorders using large amounts of data. Gene expression refers to the amount of a gene product present. The bar graph shows differential gene expression between samples A and B seen with sample A's overexpression. If sample A had a disease and the difference in expression is statistically significant, this gene can be associated with the disease. In our analysis, we are looking for differentially expressed genes between samples with different characteristics, having a mood disorder and/or completing suicide. The second analysis we performed involved identifying similarly expressed groups of genes. Similar expression implies that these genes interact with each other as part of a cohesive network. From there, we can identify how these networks relate to mood disorder diagnosis or suicide completion.
Through our 2 types of analysis, we identified genes that function in pathways related to immune system regulation, responses to infectious diseases, signaling between cells, and neural development. Targeting these key genes could lead to an effective treatment and mitigate the risk of developing mood disorders or committing suicide, ultimately increasing resilience in those that are most vulnerable to developing mood disorders. Our research is just one step to identifying major players associated with mood disorders and suicide, and helping to better understand mental health.